Hi, my name is Leah. I'm a postdoc at EBI, and I'm here to present my poster on bedaquiline resistance in South African TB. So why are we looking at bedaquiline resistance? So bedaquiline is a drug of last resort in Mycobacterium tuberculosis, which is the causative bacterium of tuberculosis. And it is used mainly to treat multi-drug resistant TB. And multi-drug resistant TB is very prevalent in South Africa, and so currently they consume more than 50% of the global use of bedaquiline. So this story really started with a data set of about 500 Mycobacterium tuberculosis from South Africa, and we wanted to ask the question of how much bedaquiline resistance do we see, and do we see it evolving clonally, or is it happening spontaneously, and can we find the genetic determinants of bedaquiline resistance? And so the first thing we did was we built a phylogeny from these 500 samples, and what we did is we then mapped the resistant isolates onto this phylogeny, and immediately what we found was that we had, based on the red arrows all around the phylogeny, we found bedaquiline resistant samples spread across the different lineages. And so this really said to us that it wasn't a clonal expansion of a single resistant lineage, it was spontaneous evolution of bedaquiline resistance. And so in order to find the genetic determinant responsible for this, what we did was we took phylogenetically related isolates that were close to our bedaquiline resistant isolates, but were phenotypically sensitive. And by doing this, we could compare a resistant sample with a very closely related sensitive sample and look specifically at the genetic determinants that were different between the two to sort of hone in on what was causing the resistance. And when we did this across our 30 samples in the data set that were bedaquiline resistant, we actually found that one gene, a mutation in one gene came up more often than not, and that gene was MMPR5, which is also known as RV0678. Now, MMPR5 is a negative regulator of an efflux pump, and it has been found before by other studies and groups. But really what is known about MMPR5 is that sometimes you get the accumulation of mutations there, but it's really unclear as to what effect that has on bedaquiline resistance, because we see this case of having mutations in MMPR5 that lead to resistance, but sometimes they don't. And so when we looked at our isolates that had resistance, I really wanted to look at what was actually happening at the protein level to this regulated gene. And so when I looked at the protein level of all of the 500 isolates for MMPR5, what I found was that from the majority of cases where you see sensitivity, you have a fully functional MMPR5, a full-length protein, but more often than not in the resistant isolates, you have a truncated MMPR5. And this truncation can happen through frame shifts and also insertion sequences, so lots of different truncations happening at different points in the protein. And so this was an interesting observation that we saw truncation, and so presumably elimination of the effect of this regulator, meaning that you got heightened expression of the efflux pump, and so resistance to bedaquiline. But we still saw this same thing that other groups have noticed, which was you can have sometimes truncation would lead to resistance, sometimes truncation would lead to sensitivity. So it really wasn't clear still, but we thought that perhaps maybe this wasn't, maybe there was an effect, but we weren't seeing it clearly with the binary phenotypes, with the sensitive resistance. So what we wanted to do is we wanted to move to a tuberculosis cohort that had minimum inhibitory concentrations. So the next thing we did was we looked at the cryptic data set, and we looked at all South African Mycobacterium tuberculosis from that, which was just over 5,000 isolates. And we went to see how many truncations in MMPR5 we could find in this data set. And it was only about 100, but we were able to characterize all the different mutations we saw in MMPR5. And more often than not, we saw frame shifts leading to truncations. And actually, by plotting it onto MMPR5 and looking at the different domains of the protein, we could see that truncation was happening quite often before the DNA binding region, which sort of supported our hypothesis that truncation of this protein was leading to inactivation of the negative regulation of the efflux pump. And so finally what we ended up doing was in this set of 5,000 Mycobacterium tuberculosis, we looked for three different genotypic effects. We looked for isolates that had a truncation of the regulator, MMPR5. We also looked for any isolates that had inactivation or presumed inactivation of the efflux pump itself. So inactivation of the efflux pump itself would mean you wouldn't get resistance to bedaquiline. And we also wanted to get a baseline by looking at no truncation in any of the isolates. And so when we found isolates with the efflux pump knocked out, we saw hypersusceptibility when looking at the average MIC. When we were able to derive a baseline by looking at isolates with no truncations in any of the genes at all. And what we ended up seeing was a shift in the MIC to resistance based on a truncation in MMPR5. And so basically what we've found now is we've seen that with truncation of this regulator, we do get a shift in the MIC. And I suppose overall why this is important is because bedaquiline as a last resort drug is important, and diagnosis of resistance rapid diagnosis of resistance in Mycobacterium tuberculosis is very important. So if we can understand the genetic determinants of this, we can more rapidly identify resistance and better treat patients with tuberculosis. Thank you.